Hi there, good evening everybody, or good morning, depending on where you are, and welcome to today's OpenFlow cytometry session. We're again going to be talking about cell sorting today. Well, you can see that splash screen there, just a reminder that this is being run as a Zoom webinar, so you're muted and your cameras are off. But if you've got any questions, please do put those into the Q&A box, and we encourage questions. Put those in the Q&A box and we'll answer those either as we go along, or if we don't cover them during the course of the session, we'll try and get those at the end. If there's any problems with things like sound or vision, please put that into the chat box. So this is the third of our sessions on cell sorting. We're going to follow on from some of the things that we learned in the first two sessions. And again, today we're going to be using the fax aria. There's a little bit of a difference today in that you'll notice that Rui is sitting in the lab. Kathy gets to sit in, if not an office, her home and help out. So actually, let me introduce ourselves. Many of you have been to these webinars before. Some of you may not have done. So my name's Derek Davis, and I am the Science Technology Platform Training Lead at the Francis Crick Institute in London. But for many years, I ran a core flow cytometry facility, and most of my job is revolving around training. And my co-presenters are, let me hand over to Kathy.
Thank you so much, Derek. Yeah. So my name is Kathy Daniels, and I am the Flow Cytometry Core Facility Manager here at the Whitehead Institute in Cambridge, Massachusetts. That's my first time saying that on an OpenFlow class. So I've been here for a little bit, over a month, and before that I was with Rui Gardner at Memorial Sloan Kettering Cancer Center. So happy to be here, and I will pass it along to Rui.
Thank you, Kathy. Yes, and congratulations again to the move. So my name is Rui Gardner. I'm the director of the Flow Cytometry Core Facility at Memorial Sloan Kettering Cancer Center in New York. And yeah, today I'll be the one operating the instrument. And I have with me Fang, who's been amazing at preparing all the samples. So everything you're going to see today has been the work of Fang. And I also want to thank Francis Weiss-Garcia for giving us the cells for us to do this class today. So thank you.
Okay, so what we wanted to do, first of all, while Rui actually prepares the machine a little bit to get ready for the first part of the practical session here today, was to give you a rundown of what we're going to do in the next 90 minutes or so. So we wanted to do a very brief recap of how cell sorting works. So many of you have been to the last couple of talks, so it hopefully will be more familiar to you now. We will continue to talk about some of the practical considerations, and this is quite an important aspect. We won't maybe talk about them too didactically, but if you have any specific questions about your cell type or anything that we talk about, this is your chance. You've got us here live for 90 minutes. Ask as many questions as you like. We want to cover also some of the recording or the records that we need to keep when we're doing cell sorting, which helps us with troubleshooting, and it helps us become more familiar with our sorters. Assuming that everything goes to plan, we wanted to put in a couple of slides towards the end about what to do if things go wrong, so troubleshooting your sorts. And the practical part of today, as Rui just mentioned, will be performing and assessing a relatively simple sort, but we're moving away from beads and from cytotrol cells or immunotrol cells, and we're using real live GFP cells today. So this slide should cover the pros and cons of electrostatic droplet deflection sorting, and that's what we're doing in flow cytometry. There are other ways of sorting cells. We can use magnetic beads, or we can use centrifugation. But in terms of flow cytometry, the vast majority of cell sorters are going to use electrostatic drop deflection, which, if you remember, is the same principle as the inkjet printer. We're taking droplets, we're giving them a charge, and we're moving them to a particular place. So in effect, our cell sorters are sorting drops, not cells. The cells happen to go along for the ride. But the great advantage of flow cytometry or cell sorting is that we can sort very pure populations. 99% purity should be relatively easily achievable, and that's generally what you want as an end user. After your transfection, not all your cells are fluorescent protein positive, we can run them through a cell sorter and give you, almost 100% pure cells of interest. And a cell sorter is no different to an analyzer in the fact that it makes single cell measurements based on fluorescence. So if you can run a 16 color phenotyping panel on an analyzer, you can transfer that panel to your cell sorter, assuming it has the same capabilities. So we can sort on as many parameters as you can put into your cell system. We can also sort on the level of fluorescence, remember, so we could sort our bright GFPs from our dim, from our negative GFPs, for example, and sometimes that's important, something that's very difficult to do with beads. All of our sorters will be able to recover more than one population. Many of them will do 2, most of them do 4. There are a couple of them that do 6 populations at once. And I would imagine in the next few years, we'll see sorters that do even more than that. And although we're not going to specifically do a single cell sorts today, single cell sorting is one of the growth areas in sorting in recent times. Many of our users are sorting one cell into a single well of a 384 well plate, and then taking that off for downstream genomic applications. Of course, there are a few downsides. Sorters are more expensive, both to buy, to maintain, and to buy time on as well. They do require a little bit more expertise. It's not impossible to train people to use a cell sorter. In fact, all of us here on this side of the call, at least, are big proponents, I think, of training end users to do the sorts if that's appropriate. But generally, it's people in the core facilities who are doing that measurement. Sometimes with cell sorting, we have to think more about our timeline, because sometimes you may require a sorter for an entire day. So you do need to plan. You need to herd your resources to make sure you can get that time. And some people are booking 6 to 8 weeks in advance in our lab just so that they can get their favorite cytometer for their entire day. And something that we will touch on a little bit later is how long it actually takes to get cells back. Obviously, all of the sorts that we've done in these webinars have necessity been pretty short. We only have 90 minutes, but you do need to know how long your sort is going to take. So here's our nicely animated slide from Rui of how cell sorting works, all in one slide. Again, apologies if you've seen this before, but the cell sorter works in much the same way as an analyzer, in that we have to prepare a single cell suspension. We hydrodynamically focus our cells through one or more laser beams, and at that point we get the information about those cells. And in order to sort them, we eject those cells into air through a nozzle in a column of fluid, and we break that column of fluid up into droplets by applying a vibration to that nozzle. We vibrate it at a known frequency, so a number of times per second, and a known amplitude, so the amount of work we get that nozzle to do. And that combination will cause that stream to break off at a very specific point, which cunningly we call the drop breakoff. And the number of drops we produce per second depends, as we've seen, on the pressure that we apply to our fluid. It depends on the nozzle that we've selected. In general, the more drops we produce per second, the faster we can sort, but that has to be tempered with the size and the type of the cells that we're sorting as well. So here's our cell. Our cell goes through our laser beams. That's the point we understand its phenotypic characteristics. We then wait for that cell to pass until it's just at the breakoff point. So it's in the last attached drop. And when it reaches that point, we apply a charge to the entire stream. And we continue to apply that charge until that drop with the cell of interest has broken off. And at that point, when that cell is now no longer attached to the stream, sorry, that drop is no longer attached to the stream, we discharge that stream. So now the stream is negative, but our drop containing our cell of interest is carrying a charge, either a positive or a negative charge. And remember, that charge doesn't affect your cell. This is a bit like this drop is like a Faraday cage. The charge is around the outside of the drop. but because it's charged, it's going to pass through this high voltage deflection field caused by these deflection plates, and it's going to be dragged off one way or the other, depending on its charge. So in that way, by putting either a positive or a negative charge, we can sort two populations, or by having a variable charge, so a strong positive, a medium positive, and a low positive, we can sort three populations to the right and three to the left. And you'll be familiar, hopefully, from our previous webinars, of this sort of picture. This is what we're monitoring in the background when we're doing our cell sort. Here's our break-off point. So we have a camera focused on this here. So this is it in real time. Here's our column of fluid, and you can start to see it breaking up. It's undulating. It's starting to break up into droplets. And then here are our detached droplets. And if you've done any flow sorting, you could look at that and think this looks lovely. These drops are nice and round and uniform. The undulations in here are fairly symmetrical. This looks like a nicely prepared nozzle. And if you do learn how to do cell sorting, you'll spend a lot of time looking at this sort of thing, but you'll understand when things start to go wrong. But the most important thing we're going to do in cell sorting is this drop charge delay, is to work out how long we have to wait for the cell to pass from the interrogation point to this point when it's in the last attached drop. And we're not going to set this up today. We saw how we did this last time, but on the Aria, we use a system called AccuDrop. So the way BD systems do it, we have a red fluorescent bead, which we pass through. There's a laser here in the cytometer, so we can illuminate both the side stream, the sorting stream, and the center stream. And what we want to do is to adjust this figure here, this drop delay, until all of our beads are being deflected to the left, and we don't see any beads in the center stream. So this is this in cartoon form here at the top. And this is the point we want to be at, because we know then that we're not losing anything that we could have sorted. Now, if we have this sort of situation where some of the beads are being sorted to the left, but some are still in the center stream, then we know, although, yes, we are sorting some of the things that we're interested in, we also know that we're losing some that we're interested in because they're passing straight to waste. So one of the things that Rui has already done is set this up, is set the AccuDrop system up already. The other thing we have to think about when we're sorting is what sort mode we're going to use, how many number of drops are we going to sort, and what is our priority? Is our priority getting purity? Is our priority getting a high yield? Or is our priority making sure that we only sort single cells? On the Diva system, and we talked about this last time, I won't go into it too much here, but we can come back to it in the session in a moment when we get to the cytometer. In the BD system, each drop is divided into 32 virtual slices. And although that column of fluid as it comes out of the nozzle is a solid column of fluid, we know exactly where it's going to break up into droplets, and we know where the cell is in relation to that drop. So sometimes that cell is going to be in the middle of the drop, which is great. If it's in the middle of the drop, it's relatively easy to sort it. But the arrival of cells at the interrogation point is a random stochastic event. And sometimes the cell might actually end up being very close to the leading or the trailing edge of that drop. So we have decisions to make, don't we? So we always consider the drop that the cell is going to be in, plus the following drop and the preceding drop. Now, if our cell is in the middle, we're probably only going to sort that drop. Okay, even if the cell traveled a little bit faster or a little bit slower than we expect, it's still going to be in the drop that we expect it to be in. But if the cell is towards the edge, then we have to make that decision. Do I just sort this drop? Or more likely, I will sort both of these drops, because that will give me the best chance of capturing this cell. And there are different sort modes, and we'll look into those in a little bit more detail in a second. And we can adjust those depending on what our question is. So most people would go with this one, the purity mode, because that's what you want from your cell sort. You want a pure population of cells of interest. But from a sort operator point of view, actually the yield is probably more important. We want to be able to give you as many cells as possible back from your tube. If you've got a 10 million cells, of which 10% are GFP positive, we should be able to get 1 million cells. As we'll see in a moment, there are reasons why we can't get that, but we want to try and get that as close as possible. So thinking about these sort modes will help us do that. So if you've got any questions about that, please put them in the Q&A, but we don't want to spend too much time on these slides. We want to go to the ARIA. So we're going to go over to Rui. Hopefully he's still there. I'm going to stop my share and we'll go back to the ARIA and we will see the setup for this sort.
Okay, thank you, Derek. Wonderful. So let me start by sharing my screen here and I can show you what we're going to do. Okay, so the first thing is what we're planning on sorting is it's a breast cancer cell line. To be more specific, Francis is just texting the actual cell line. It's an MDA MB231 if you're curious to know what we're going to sort today. Now I've cheated a little bit here where I've run already the controls and now I'm going to load the sample and we're going to gate based on the controls very quickly. So if you have any questions, just let us know. Put it in the Q&A. Because I'm going to go a bit fast in these parts because we've done this already previously in other sessions. Okay, so one of the things that's important is for us to start always at the lowest flow rate as possible because that's where we get the best resolution. So we usually always start with a low flow rate, and then we see as we start sorting, we know what the maximum number of events that we can run. And I'll explain that a little bit in a moment. And we adjust the flow rate to reach that maximum. So I'm just going to run a little bit. So all the settings have been optimized. And now all I'm going to do is record this. Okay, so recording the sample just for us to be able to set the gates appropriately. And again, since we're using the controls, we didn't really need to run the sample before to set the gates. But I'm just moving forward with this. Okay, so here's the unstained, right? So I'm going to start by gating very quickly the, trying to remove a bit of the debris that will be in this left lower corner. Now notice that actually the control, the DAPI and the unstained control are actually slightly different in terms of scatter. So I won't be too concerned because I can adjust the scatter later. What I'm really concerned about is to make sure that the signal is the same, right? And so I can gate these. So, oops, sorry. I'll start with this. So I have this gate here, and then within this gate P1, I'm going to add another gate for double discrimination. And I was lost here because there are things coming in from Zoom. So here we go. As you see, this is slightly, you can be more stringent or not, depending on how important it is not to have contamination. So you want to make sure that you only collect singlets. But this is probably the most difficult or subjective gate that we do. It's based on the knowledge that we have. So we've looked at under the microscope. Most of these cells are singlets, and actually we went through a lot of troubleshooting with Francis and Fang and Kathy to be able to get this cell line into a single cell suspension. Okay, so I'll continue with my gating hierarchy. So now,
sorry, Rui, can I just ask a question?
Yeah,
there,
go ahead.
There. If you've used side scatter width and height there, do you not use the forward scatter area height, area width?
We do. We usually
found that side scatter is best.
Exactly. So we usually do, and for the sake of simplicity, we're just using, because we reduce the number of plots here. But we usually do, and I think that's what here we are also using side scatter only. Yeah, so that's a good point. And actually, we showed in previous sessions that we were using side scatter and forward scatter to narrow down the singlets, right? To help narrow down and just collect the singlets. Today, for just simplicity, we're using just the side scatter. If you ask me which one is better, I don't think there's one better than the other, and that's why some people use one or another, or use both, which is what we do. And actually, I usually cite this guy called Derek that I remember once you told me that you used side scatter, I don't know, side scatter height versus forward scatter area, something like that. So you used a combination of those.
I tend to use forward scatter height, forward scatter area, and side scatter width, which works
Right.
quite nicely on the BD machines, because
Mhm.
it also
Right.
depends on the geometry, obviously, of the
Exactly.
optics in there.
Right. So the so today for simplicity we're actually just using side scatter, but you're right, we could use really anything that we want.
Yeah.
I don't know if you
I
wanted
think,
to say something, Kathy.
yeah, I just wanted to say I think it's a good thing for everyone to be aware of all the options as well, because it's a really good thing to explore based on your specific cells what has the best resolution. So sometimes you might have one set of cells, and it does not necessarily mean that that gating strategy is going to apply to all.
Absolutely, you shouldn't always believe everything we say. You should explore your data.
Exactly. Especially when it's me saying it. Okay, here we go. So we have the gate set based on the unstain, and now I'm going to check on the GFP control. And as you see, we are excluding the negative populations here. Now, in sorting, we tend to be a bit more, especially when the populations are very close, we tend to be a bit more conservative, and to make sure we don't have negative cells being captured by the positive gate and vice versa. And for now, we're going to sort just the GFP positive, and we're going to do a sort in purity mode and in recovery mode or yield mode, and we'll see how it goes, and compare the two. So I just draw all the gates based on the fluorescent fluorochromes that we have. So we have DAPI for live dead, as you see here. And we have GFP, right? So I'm going to the DAPI control just to make sure, and this makes sense. So we're excluding all of the positive events. And here I should be just showing what is in P1, right? So I'm going to show P1 only, right? And this makes already a bit more sense. And here I'm just going to show P2. This is P2, right? There we go, P2. And again, you may want to exclude these because they may be cells that are dying. But typically, even cells that take in DAPI or PI, they tend to they may grow afterwards. So it depends on the downstream applications how clean you want your sort to be in terms of cells that may be dying or not. So it's really up to you. And then here, here I have just DAPI versus. Oh, actually, this is the same plot that we have, DAPI versus forward scatter. So we'll just take that out and leave this here for the sake of convenience. So I've used the controls to set the gates, and now I'm going to check the sample. And as you see, oh, here I should just put P3, right? And there we go. So now we're excluding all the doublets, and we're excluding all of the dead cells. And now this seems a bit more clear. And I'm going to apply bioexponential just for us to see what exactly are we gating. Now, you may want to, when you set it to bioexponential, you may want to make sure that you're selecting. So do you see here? This is the way that Diva is transforming this gate when it goes to bioexponential. So I'll try to just make sure I select. Oh, I should be. Oh, right. This is single stain, and I'm going to the unstain just to make sure that this gate makes sense and we're not capturing any of the negative cells, right? So okay, so we have the sample here. It's up to you if you think you're missing on some events. You can be, again, more or less stringent. It's really up to what you're doing downstream of that, right? So for simplicity, I'm just going to leave it like this and making sure that I'm not collecting any of the negative cells. Okay, and so what we're going to do now is we're going to sort in purity mode, and then I'm going to sort in recovery mode, and then we'll do, so then while I'm cleaning the system, Derek will show you some more information on purity versus recovery. So here, let me see. We're going to set up for global sheet 1, we're going to set up the sort layout. So I'll bring it down here. And again, if you have any questions, just let us know. I'm going to set on the right, I'm going to set the population that we're going to sort, which is P4. And now here, we're going to choose, well, we can have a 2-way, a 4-way. In this case, I'm going to leave it in a 4-way because we are going to do 4-way sort. Yeah, Kathy, go ahead.
Yep. So I just wanted to interrupt quickly because we did have a question asking if you always have the gates extending off the plot. So I think they're referring to P4 and how it's going completely off scale with the gate.
Okay, that's a good question. So what we do usually, if we want to capture everything, because sometimes things are squashed in the axis. So to capture those events, we go, like what I'm doing here in P3. Do you see that there are events here squashed in the axis? To make sure we're capturing all these, if I transform here by exponential, you'll see that there are many more events underneath, right? But if I put this gate where it was before, then now, if I turn by exponential again, you see that now it's capturing everything, right? Again, when you do funny things like a diagonal gate, it's better to look at the gate in by exponential, just to make sure it's not doing funny things, like you're seeing here, and make sure it's not capturing actually negative or other events that you want to exclude. But in this case, for instance, in DAPI, we don't need to, because we're excluding, we're getting everything that's negative for DAPI and excluding everything that's positive. So we leave it like that.
Great. And then we do have one more question that came through asking a very important question, I think, about whether or not we can gate on histograms for sorting. And I think I would expand on that question to say, is it can we, and then is it recommended?
Right. I mean, you can, of course. But the thing is, we want always as much as information as possible. And so, the more dimensions we gate on, the better we will be to not only exclude things that we don't want, but also to include the things that we actually want. So we tend to take advantage of other fluorescent channels to look at autofluorescence, and that actually helps us to pull out different populations rather than just using one dimension. But you can of course, you can use just one dimension if you want.
Okay. And there was another question there, which is not directly related to sorting, but we'll answer it now while we've got the chance. But and they're asking what are the considerations to avoid GFP quenching if you're using fixed cells and performing flow cytometry? So obviously, if you're using fixed cells, you're probably not going to be sorting fixed cells. Sometimes there are reasons for sorting fixed cells, but GFP in particular is quite susceptible to fixation, particularly alcohol fixation, because obviously GFP is a protein, and alcohol changes the conformational structure of proteins. And very often, if you fix your GFP expressing cells in ethanol, you lose the fluorescence, and you lose it because the structure of GFP, the amino acids that are close together, move apart and they no longer fluoresce. So, which is why you can buy anti-GFP antibodies. So if you're going to fix your cells that are GFP expressing, you would normally use something that's not going to change the conformation of the protein structure, which would be an aldehyde, so formaldehyde fixation of whichever strength you want to use, anywhere between sort of 0.5 and 4%. And it may be an experiment that you have to perform to work out what's the best, which gives you the best resolution post fixation, because fixation as well, of course, is going to affect your cells and potentially will increase autofluorescence. So that may be something that you have to experiment with. I hope that answers the question.
Yeah, and Derek, I'm just going to supplement that quickly to let everyone know. I'm going to put a nice reference for that in the chat function so that everyone can take a look at a paper that discusses the impacts of fixation on fluorescent proteins.
Right. And in fact, yeah, and sometimes it may be difficult to be able to, if you're trying to sort for a nuclear protein, and you need to permeabilize the cells in such a way that for the antibody to reach the nucleus, sometimes that will that permeabilization will interfere with the GFP signal, so you may lose any fluorescent protein signal. So sometimes it's a balance between trying to reach where you want to and getting that good signal. So I'm going to start sorting, and as you see, so this is a rather difficult sample to sort, and that's one of the reasons why we're using this. As you see, the number of events does oscillate a little bit because these cells tend to aggregate a lot. So we sometimes have to unload it just to vortex because the automatic shaker that's being used is not enough. So I'm going to start sorting, and you see that the numbers here are jumping quite a bit. And let's see how this works. Oh, so I was mentioning I'm going to use the purity precision mode, and I'm going to start sorting the P4. So I'm going to click sort. It's going to ask me it's going to remove the waste drawer, and so that the cells will fall, the drops will fall in the tube, and it will turn on the voltage plates. So we start sorting. We start sorting, and let me see. I'm going to just display more events so that we can see it better. And so we're sorting P4, and you see that we have here some events going through. So these are drops. And so we have a laser that's shining the waste stream. So these are drops that are going to the waste. These are the drops that are being deflected. Now I'm going to try to increase just the flow rate a little bit. So I'm increasing from 1 to 4, and you see that the number of events will start hopefully increasing a bit. Now, important things to monitor here are the number of events. So what is the maximum number of events that we can sort at? So we're using 100 micron nozzle, which is at 30,000 drops per second. And so we can sort up to 1/4 of the number of drops per second, which means 7,500. And we're pretty far from that. So this sample is quite diluted, but it's actually diluted on purpose because we have a hard time being able to prevent cells from aggregating in this case. So some samples need to be highly diluted. And so as we increase the flow rate, what I try to keep an eye on is, especially in the forward scatter, making sure that we still capture everything in the forward and side scatter, that we don't lose, because we're losing resolution, especially in the parameters that are in a linear scale. And we need to keep an eye to see if we maintain this resolution as we increase the flow rate. So I'm increasing the flow rate to 7, 8, and I still see that we can pull apart these two populations. So you see, we start seeing more of these drops being deflected, and we're actually sorting at 200 events per second, 100 events per second. And now, like I said, the important thing to take into account is the efficiency. So this gives us a very good sense of how good this single cell suspension is, and also if there's any interference, either from the instrument, but mostly from the sample, that will be, causing this loss of efficiency. What is the efficiency that we should expect? I would say anything between 70 and 85% is a pretty good efficiency. In this type of cells, we don't expect such a good efficiency, unfortunately. But you're seeing here that since the number of events are oscillating a bit, it's also hard to say exactly what the efficiency is. But it's I would say it's oscillating between 50 and 80%, right? So it's difficult to tell, but I think it's more on the 60s and 70s than anything else. Yes, go ahead, Kathy.
So we have two questions that actually came through. So as you're sorting, it's a good time to ask. The first question that came through is someone who's asking for the difference between purity and 4-way purity sort modes. We might get into that a little bit later, but just if you wanted to give a brief explanation of that.
Between purity and purity?
And 4-way purity.
Yeah. So yeah, very, very quickly. So purity mode just looks at the adjacent drops or the close drops to see if there's a non-target cell that's very close that may end up in the drop once it's charged. So so this is the concept of purity. Recovery or yield tries to sort all of the cells. So if your target cell is actually in the edge of the drop, so to make sure that we don't lose it, it will sort 2 drops to make sure that even if that cell ends up in the adjacent drop, it will be collected. So the difference between 4-way purity and purity is that the 4-way purity is just a pure purity mode. I'll pass the punt. And the purity mode actually tries to do a bit of both. It tries to capture, tries to eliminate those non-target events, so it doesn't sort the cells when a non-target event is close, but it also tries to recover as much as possible. So it will try to sort 2 drops as long as there's no target event close, or even close to that adjacent drop. So it's just a better mode to try to recover as much as possible. The reason I'm using purity mode here is exactly to be able to show you the difference between that and recovery mode.
And I think it's a nice added benefit, I think, to tell people as well that if you're very cognizant of the volume that you wind up having to sort, you have to be mindful that, you know, if you want to make sure that you're only selecting 1 drop, you be mindful of your sort purity mode as well, especially for sorting into trizol or anything where volume's dependent. And then we have one more question that came through that was asking if you can sort with bi-exponential axes on the plots.
Yes, we can sort with bi-exponential. The only thing we need to be careful with is that the gate is not below 0. So all of the gate is not below 0 in any of the parameters. This is a limitation of Diva. It's not necessarily, you won't see this most likely in other instruments. But this is again, this is a limitation that Diva has. So you just want to be careful with that. But you can sort in bi-exponential, yes. So I've sorted in purity mode, and we sorted 27,000 events. And now I'm going to sort in recovery mode, and then we'll see later the difference between both. Okay. Okay. So I unloaded the sample just because, like I said, the cells tend to aggregate, and then it becomes more difficult to sort the singlets. You're going to start losing cells because most of them are aggregating. So okay, here we go again, and now I'm going to switch to recovery mode, and you're going to see the difference between the efficiency. So I go to yield or recovery mode, and I'm going to start the sort. So I change the collection tube, and here we go. So we're sorting. Now again we're about 1,000 events, so it was between 1,000 and 2,000 sometimes. We're sorting now a little bit more events per second, and look at the efficiency. The efficiency is 100%. Why? Because it's not aborting anything. It's just trying in this mode. We are just trying to sort all of the cells as possible. Now, if we have non-target cells close, then it's going to contaminate, and so you'll see most likely purity is not going to be very good, or as good as in the purity mode. But the efficiency is close to 100%, because again, it's not aborting anything based on your non-target or target cells, right? So I'm just going to go and sort. So it's going much faster, of course, and that's why you have to take that into account when you're sorting in purity mode, that you're going to lose some cells. So if you have a target number of cells that you want to sort at the end, you need to consider that you're going to have an efficiency. It could be 70%, could be 80%, or it could be 50%, as we were seeing here, oscillating, right? So I'm just going to sort more or less the same number of cells, and then I'll pass it on to Derek. Is there any other question or any other thing that you want me to show here? Trying to think of other things that could be important.
Well, there is a question about efficiency, but I think I'll cover that in the slides in a moment.
Okay.
I mean, you mentioned the efficiency there, and as
Okay.
you can see, there's a big difference between the efficiency when we do a purity mode sort as opposed to a yield sort or a recovery sort.
And watch the zero events being aborted, right? Okay, so I'll stop here, and then I'll pass it on to you, Derek.
Okay. I need to grab the screen share then. Right, that's it. There we go. Amazing how silent it goes when Rui switches, goes on to mute, isn't it? You don't think there's too much background noise, but it's not too bad, but you notice it when it stops. Anyway, so what we wanted to do in this bit, while Rui cleans the machine, so remember we always want to check the purity of our sorts after we've run our sort, because that shows that the machine has been doing what we want it to do. So in order to do that, we need to clean the machine. So that's what he's doing at the moment. And I said we'd talk about a couple of specific practical considerations, a couple of which have already been mentioned as we've gone along. And one of them is this whole concept of aborts or losing cells that we could potentially sort. And this is quite important in sorting, particularly if you're sorting a rare event. You want to make sure that you give yourself the best chance of recovering all of those cells. You don't want to lose them for reasons that we could avoid. And there are two main areas where we lose cells or we fail to measure them or fail to sort them. And we divide them into what we call hardware or electronic aborts and then software or coincident aborts. You might hear them referred to as other phrases in other sorters or from other people. And by hardware aborts, what I mean is that I've got two cells that are so close together that the cytometer can't measure both of them at once. Now, this is much less of a problem with modern flow cytometers than it used to be. So nearly all of our flow cytometers are now analog cytometers, digital cytometers, as opposed to the analog ones that we had 20 or so years ago. And with an analog cytometer, if you had one cell that came along and then another one that came very quickly behind it, the cytometer electronics were tied up measuring that first cell and couldn't measure the second one. So what that would mean is that you wouldn't know what that second cell was. So we wouldn't sort either of those cells because we would risk contamination. Now, with our modern digital machines, it's much easier to measure cells or events that are closer and closer together. Not completely impossible for us to miss cells like that, but it's a very low level. And if you were watching those electronic aborts, you would see that was a relatively low level, a few events per second when we're going through it, several thousand events per second. More likely, we're going to lose cells by what we call coincident aborts. And this is, I've got two cells that are sufficiently far apart that I can make individual measurements, so I know what those cells are, but they are going to end up either being in the same drop or they'll be very close together in adjacent drops. And as we've seen, we can adjust the way that we run the flow cytometer to make sure that we don't lose any of those cells. Now, if I've got a drop that's got a target cell and a non-target cell, if I want high purity, I'm going to abort that. I'm not going to sort that cell because my purity would be reduced by that non-target cell. But if I'm running in yield mode, recovery mode, I will sort that because I'm more interested in getting the target cell back than a little bit of contamination with a few non-target cells. So we have some control over that one. But we're always trying to make sure that we use our knowledge of the way that the sorter is sorting and the biological question that we're asking to maximize our chance of being able to get as many cells back as possible. And it always comes back down to this, doesn't it? The sample, your sample preparation, as we've said. Sample preparation is key in any flow cytometric experiment, but it's even more key in a flow sorting experiment than it is in an analytical experiment. Not just because we run the risk of blocking a nozzle in a sorter, and that's a bad thing, but the more spread out, the more monodisperse our cells are, the easier it is going to be to sort them individually. So another question that comes up and that Kathy touched upon just now is, or one of us touched upon, is how long you need to actually sort your cells. And this is an important consideration. Now, if I'm sorting, the number of cells I can sort per hour or time period depends. It depends on the pressure I'm using. It depends on the nozzle that I'm using. But if we take a generic 10,000 cells per second, whether it's 100 or a 70 micron nozzle, that means that I can run my cells through at around about 35 million per hour, something like that, which sounds a lot, doesn't it? But sometimes you're looking for a relatively small population. So if you're looking for example, at a 1% population of GFP positive cells, and you need to get 100,000 back to do your assay, that means you're going to take 17 minutes or so, around about 20 minutes of sorting. That sounds pretty reasonable. But if that population was much less abundant, and that's not particularly rare, is it one in a 1,000, for example, but now you want 1 million cells to do half a dozen microarray experiments, now you're going to be sorting for 28 hours, and neither you nor I want to do a 28-hour sort. So knowing something about the biological question and the target frequency is going to be important in helping you decide how long you want to book the machine. Now, in this case, we would work with you to do some sort of pre-enrichment or book half a dozen cytometers to get that done much more quickly. And of course, even if you're in this situation, if you say, I've got 10 samples that I want to sort, we're not going to be able to do that in an hour or 2 hours or even 3 hours, because remember, we have to factor in time for cleaning in between. So all these practical considerations are quite important when you're thinking about actually turning up in the flow lab. And the other thing that Kathy definitely did mention is the volume of the fluid that we're going to get back. And that is going to vary as well, depending on the nozzle size. Obviously, the bigger nozzle is going to produce bigger droplets. And the actual volume depends, again, on the frequency, it depends on the pressure, and depends on the size of the nozzle. But as a rule of thumb, again, this is sort of a ballpark figure. This is taken from the Sony, the SH800 machine that we used in the first of our demos. If you're using a 70 micron chip, then the drops that are producing are around about 1.5 nanoliters. 100 micron, it's about 3.5 nanoliters, and 130, it's around about 7 nanoliters. Now, this becomes important because sometimes we're asked to sort into very small volumes of trizol, for example, 2 or 3 microliters. I only have to sort 1,000 of these cells, and suddenly I've doubled the amount of fluid and therefore halved the concentration of my trizol. So it's important to think or remember that your cells are enclosed in a droplet of fluid, and that's going to contribute to the volume, your sorted volume. It also has a practical implication, how many catching tubes do I need to bring along? And I know that with a 70 micron nozzle, I can normally fit around about 5 million cells into a 5 mil Falcon tube. With 100 micron nozzle, it's about 100. It's about 1 million or something. You soon get used to that. One thing that you might have seen pop up at the end of that sort was an alert from BD to say, do you want to save a sort report? Okay, and that's quite an important thing for us in the core facility, because you're coming down to the lab with a reason to sort your cells, and that reason could be many and varied. You want to regrow them or transplant them or just look at their morphology or something. Obviously, we've got an output in the fact that we were generally going to save an FCS file of your sorted cells and your pre-sorted cells. But there are other metrics, as we've seen, that we quite like to get a handle on. What was the frequency we ran? What was the pressure that we were running? Which nozzle did we use for this experiment, and so on? And all of those things help give us confidence. And the more we know about what we've given you, that also gives us some sort of insurance policy. If you come back and say something has gone wrong, it gives us a starting point to troubleshoot. And some cytometers do this automatically. So this is an example that you definitely won't be able to read at all here of a sort report from the DIVA system. So it will tell you the frequency and the amplitude that we used. It tells you the drop delay that we settled on. It tells you the precision, so the masks that were used. It also says how many events were acquired. So you can see here that we acquired 141,000 events, but there were only just over 1,000 electronic aborts, so very low abort rate. And then it tells you something about the actual sorted cells themselves. So we run something like this, but not all of our machines are DIVA based. So we also have a Filemaker Pro database that has additional information. So it contains things like the fluorochromes and the voltages that were applied for each of the parameters. It tells you something about the experiment, and we've got space in there for putting in screen dumps as well. And it's also got an audit trail, so we know who actually ran this sort and where they came from, who did the sort, and so on. And it's always very useful to have those pieces of information. There's a question there actually from Sarah about how do you get that nice looking table instead of the one that looks like embedded in code. So do you mean that table?
Yeah, I'm thinking that Sarah might be talking about when you go to the D drive and go to the reports like that, it doesn't show up as a PDF like that. So it has to be at the time of the sort, right? Where you're clicking on the actual sort layout and then go to the sort report from Diva at that time.
Right, yeah, you have to actually physically tell it to create the report, yes.
Yeah.
Okay, so hopefully that's given Rui enough time. So we'll go back to him, hopefully he's still there. Well, I've gone off for a cup of tea. Oh, no, there he is.
Sorry, I had on mute. Here I am. Yes, I haven't left. So I was so I'm just finishing recording the purity mode here. And I'll share the screen while we're doing that. That would be a good idea, right? Oh, overwrite. Okay.
Yeah,
sorry,
otherwise
guys.
you're going to have to do it in MIME.
Exactly. Why am I'm trying to find the, I can't find the place to share the screen. Oh, there you go. I'm in the minimized mode. I was looking for the button to share the screen. Like, I'm a flow cytometrist, not a zoomist. So I'm good at sorting, not doing Zoom calls. Okay, so here we go. I just okay. So what do we have here? We have the sample that was sorted in purity mode and the sample that was sorted in yield mode, right? So I recorded what we sorted, and you can see that we have here, if we look at the gates we have, so we added DAPI to the post-sort sample just to make sure that we would exclude any dead cells. But if I plot here, just instead of P3, if I show P2, which I will do like this, and we can see, for instance, that let me just copy this gate and paste it here, and so if we paste that, then I can see that the the purity, if we exclude the dead cells, is slightly better, right? It's 93% instead of 92. So and that's one of the reasons why it's nice. It's a nice idea to add the viability die in your post-sort sample, because it helps you to determine whether some of the events may have been positive events, but there are cells that just died, and so end up skewing a bit the purity. And I think you're going to talk a little bit about that, right, Derek? And then, but so what we need to compare really is this percentage, right? 93.7% with the same gates. Oh, and I should say that when I recorded these, I recorded exactly at the same flow rate, which was 1, that I recorded the original sample. So if you want to compare things, you need to compare apples and apples. So if you've run your sample, you recorded your original sample at flow rate 1, you need to record also your post-sort purity checks with the same flow rate, in this case flow rate 1, or else you may lose resolution if you're running it higher, and so of course the population will move away from that gate, right? So we need to be careful with that. And so comparing purity mode with yield mode, we see that, okay, it wasn't bad actually in yield mode. We had 89% compared to 93.7%, but and so it shows that we were still able to get, if 89% for you would be fine, that would be okay. One thing I usually say, and I don't know if I mentioned in other classes, is that with a droplet cell sorter, we should expect 99% purity or more. So if it's not 99% purity, there's something wrong either with the way the instrument was calibrated, or with the sample. Now we know that this sample is relatively hard to to gate out the doublets. So there's a good amount of heterogeneity in the actual cells if you look at the original sample, and and we're trying to exclude what we believe are doublets, but we may be still including some doublets, right? Depending on how we set the gate here, we may be still including. So if you're not happy with its purity, then it would be important for you to exclude even more, to be more stringent here in the doublet discrimination plot, right? And as you saw, we sorted much faster in yield mode. So for instance, if 90% purity was fine for you, then um, then then you could sort in yield mode, right? But the only caution here that I should mention is, if you're sorting in yield mode, the reason why we didn't see a huge difference between yield and purity is because the sample is very diluted. So if the probability of having a non-target, or even any cell, right, not only a non-target, but the probability of having any other cell together is going to be reduced if the sample is very diluted. With a more concentrated sample in yield mode, you would see this purity drop quite dramatically, right? So
Yeah.
just to mention that also.
Yeah, actually, I was going to come back to that because I forgot really to talk about the efficiency in those slides
Okay.
that we saw, didn't we? When we changed to the yield mode, the
Okay.
purity went the efficiency went to 100%.
Mhm.
And as we said, because it'll always sort the cell that you want it to sort, no matter whether there's anything else nearby or not.
Right.
And when we were in purity mode, that efficiency dropped because there will be occasions where you've got a non-target cell in the same drop or nearby. So therefore, the efficiency is giving you a readout of how efficiently the sorter can sort out the cells that it wants to sort, if you like. And I never get too hung up with that, because you could sort at a very low efficiency, if you like. You get very high purity, but it'd take you a long time. So you're always going to have this sort of balance between how long do I actually want to spend doing this sort, not just from your point of view, but from the point of view of the cells. I always try to get my cells in and out of the sorter as quickly as possible, because we don't want to induce any damage or anything untoward to the cells. So the quicker we can do that, the better. So again, it's all about the knowledge, isn't it? Having the knowledge of what the effects of doing these things are.
Exactly. Absolutely, I agree. Do you want to show the slides while I'm setting this up? You can go ahead.
No.
Okay.
No.
Okay.
No. This is what it's like at the sharp end, Rui. You see, this is what Kathy's been doing for months.
Exactly. Okay. So what I'm going to do now is I'm going to sort in purity mode, right? And I'm going to sort essentially, the same. Here, let me take this out of the way. We're going to sort now the same two populations, so positive and negative, but based on the singlet and the doublets. So we'll try to compare and see, what would be the difference in these two populations if we were including more doublets or less doublets. In this case, this second gate is essentially just doublets, right? So, it's going to be hopefully pretty obvious. So we haven't tested this, but hopefully it will be pretty obvious when we compare both. So I'm trying to be stringent here with the singlet gate, and I'm going to try to exclude here the DAPI positive cells. So I almost should be using the same gate. And then here, I think these gates are fine. I'm actually going to bring the negatives a bit closer. So essentially, these are the same gates, right? I mean, I could copy paste and do exactly the same gates, but now for the sake of comparison, do you see that even the autofluorescence, and we're using the P channel again as an autofluorescent channel. We could do it in a histogram, but actually, why don't I just draw a plot here, a histogram, for you guys to see what would be the difference? So if I would, let me see, if I would put GFP here, and here I would just look at the live cells, right? And this would be my GFP versus so negative versus positive. Now, as you see here, it may be, depending on the overlap that you have here, it may be difficult to gate out just the positives. Whereas here, when we use autofluorescence, again, I won't go into details, but PE here is not the best parameter. This classically, PE was a good parameter because we were looking at PE excited by the blue and looking at the PE emission filter, and that's where you capture a lot of autofluorescence. But PE now we use the yellow-green laser, and in that, the yellow-green laser is not as efficient at exciting autofluorescence as the blue laser. So, but still, it ends up being a bit easier to, especially if you see these diagonals, we're able to set these diagonals in a way that we can exclude and have a bigger gap between these two populations than we would have if we were just sorting based on a histogram. But you could do that. I would recommend always using more dimensions. Of course, we can't gate in 3 dimensions or 4 or 5. So we're stuck with an extra dimension, but it does help us to exclude things that we don't want. Okay, so I'm going to sort these samples, and then we're going to see how it works, and then I'll pass it on to you. So I'm just going to add the collection tubes very quickly to start sorting. Okay, we're using Eppendorf's just because it was easier to check purity. You can use whatever you want. You can use any type of tubes that you want. It really depends on a lot of things. How many cells you want to collect, and also if you want to check purity, you can always remove an aliquot of the sample, so you don't have to sort in Eppendorf's. But we did do that here just for the sake of simplicity, that we don't have to be aliquoting anything. Okay, so I'm going to load the sample again. Oh, so what I'm going to do here is I'm going to sort P6 and P5, so GFP positive and negative. In these are in the where's the hierarchy? Here it is. These are in the singlets, and then the P8 and P9 are in the doublets, right? Okay, and so one important thing is when you're sorting in 4-way, you want to have the more frequent populations in the middle and the less frequent on the sides, right, on the far end. And this is because if you're sorting the more frequent populations on the far end, you're going to have more drops, and the possibility of those drops eventually also falling in the inner tubes is going to be higher. So you want to switch that and make sure that the more rare populations are sorted in the far end. Right. Actually, Fang is giving me an important thing. We would never be able to sort here because I'm using a different global sheet. I'm using global sheet 2, and this is for global sheet 1. So thank you, Fang, for that. That's why it's very good to have Fang here while we're doing this live. So I'm opening this. And in this case, we use 4-way purity because it's a more efficient precision mode when you're sorting 4 populations. Because remember, it has to charge and uncharge all of these different drops depending on the cells. So we're sorting 4 populations, and if we're using purity mode is trying to sort the second drop if there's no contaminant. So you would have many more aborts if that was the case. And also you have more fanning. I won't go into details, but you would have also a little bit more fanning with that case because it's trying to charge more drops. Okay, so what we do is we look at the percentages. So for instance, here we have the total percentages: 35%, 7, 14, and 0.8. So what I'll do is I'll put P9 on the far right and P8 in the right. So I'll add P8 and I'll add P9, and on here I'll add the P6 in the far left, and then I'll add P5. Okay, 4-way purity, and I think we're ready to go. Right, we're ready to go. So I'm going to load the sample, And I'm going to start running it, hit sort. When it loads. There we go. So the sample is running. I'm running at, again, 1,000 events roughly, with, and I'm going to start the sort. Yes, here we go. I'm nervous and excited at the same time. Okay, so here we go. We see that we have 1, 2 populations, 3, and once in a while, it may be difficult for you to see here, but once in a while, we'll see a little flash here. You see there? It may be difficult to see. You can try and adjust the laser. So I'm moving it. There we go, a little bit more. But it's sorting just 16 4 events per second. So we're going to have very few events of these. Whereas with the others, of course, based on their frequency, they're going to be sorting much faster. So this one will be less. And you can see now that, the efficiency looks more like what we're used to, 72%, 81%, 74%. Now this one, 33, 40, 80, this one is going to oscillate much more, just because statistically we have very few events. So it's trying to calculate an average efficiency, but it's sorting too few events live, and so it won't give us a very accurate number as it's refreshing. So it becomes a bit harder to interpret. Like now it seems that I have 100, but then I have 0. So it depends on how the amount of events that are being sorted. And again, just like Derek mentioned, you have the conflict count and the conflict rate. So this is basically the number of events that are being aborted, which could be hard abort versus soft abort. But we're not, where do we see here the, I thought we could see here there.
Yeah, the one on the left, yeah.
Exactly. Thank you. So here we see the electronic abort rate, but it's very low compared with the amount of events that are going through. Like we're running at 750 events per second, and we're seeing maybe 24 events per second being aborted.
The one thing here, Rue, we've got the 100 micron nozzle, and we've got the sweet spot on, haven't we?
Yeah.
Okay, so the sweet spot from the last time is it's automatically adjusting the amplitude to try and keep that break off point in the same place, and that's quite a useful sort of catch all, isn't it, when you're sorting?
Right. You mean having the sweet spot on?
Yeah.
Mm-hmm. Right. If we don't, we could control it manually, right? Here it's pretty stable, so it's been at 4.6. The amplitude, but yeah, you could adjust it manually, like
I think those of us who have been in the game for a long time, when something automatic like that comes out, you think, well, I don't really trust that. So you have to get to the point where you do trust it to do what you wanted to do. And as you say, like in the early days of Flow, we would be watching that the whole time, adjusting the amplitude minutely to keep that in the same place. But now technology has moved on a little bit, and it'll do that for us.
Mm-hmm. Yeah, exactly. It's the more, the more things develop, the more you start trusting them. I remember also, being very nervous with machines like the Arias, because it seems like a black box that you couldn't do anything, and with the other machines you could control a lot. But to be honest, now I see the newer machines that look like a black box, and the Arias, I feel like I can do a lot, right? So, because you start trusting the machine.
Yeah, it's like when the internal combustion engine came along, Rory. I remember that.
Right, I don't remember that, Derek.
I definitely do not remember that, but we do have a question that came through. So someone had asked, is it okay to adjust that AccuDrop laser, the diode laser during sorting? They thought that this was adjusted for the AccuDrop calculation itself, and therefore should it not be changed during a sort?
Right. Yeah, this is just a laser, so it's not measuring anything now, right? This is just a laser for us to monitor the quality of what's sorting. You see that there's some fanning, especially in the ones that are being sorted in the far end. And so this is just again, it's a laser that's beneath the plates, and it's not measuring anything at the moment. It is used to measure the AccuDrop, so the timing between the lasers and the breakoff point. But so during that, when you're measuring, you want to fine tune it in such a way that you maximize the number of flashes, right? The fluorescent signal that's coming out of those drops and those beats. But once you've measured that, yeah, you can adjust it, freely, because it's not interfering with any measurement. Okay, so I don't know if you want to show any more slides, Derek, or answer more questions. I'm just going to leave it a little bit more because we still have only 1,000 events here. So I'm going to leave it a little bit more just to see if we can reach, I don't know. Well, actually, no, I can stop now and let's see if we can measure something out of here.
I've got a few last slides to show, and obviously you want to do a quick clean.
Yeah, exactly.
To do that. So.
And if anyone has questions about cleaning, I can also answer that later.
Yeah. Sorry, go ahead, Kathy.
Sorry, I just want to bring up one thing before Bree stops sharing his screen quickly. So, it's really nice with the sweet spot, going back to earlier, that it adjusts that amplitude to make sure that it's staying with the correct gap and everything like that. But one thing just to be mindful of and make sure you're mindful of is that drop one position, because that drop one position can change somewhat drastically over time with temperature changes, humidity changes, or, something might have happened with a partial clog. And while the instrument is pretty smart, we do want to monitor that drop one position to make sure it doesn't stray too far from the path where we originally had it, because that might require a recalculation of drop charge delay. I typically, if it goes more than about 8 or so.
Yeah, that's a good point you bring up there, Kathy, about the temperature as well, because the temperature will affect the drop break off as well. The viscosity of the fluid will change. I mean, the Arias is pretty well temperature controlled, I think, but there are other machines that are maybe a little bit more open. That is a consideration.
Exactly. Yeah, and just very quickly, I also, one thing I didn't mention is we also keep an eye on how the signal, to see if it's stable, if it's not fluctuating a lot, because sometimes that can be an indication of some fluidic problems. So having these, profiles, these populations nicely stable is good. One thing that you can do is add a plot with time. So any scatter or fluorescent parameter versus time to monitor it as it's going along, and to see if there are breaks or oscillations. That will give you an indication of fluidic problems. But with Diva, actually, it's not very easy, because it just shows you, I don't know, 1 second or 2 seconds at the time. So it's hard to tell, until you finish recording. It's hard to tell if it's actually stable or not as you're acquiring. But you can always watch it later, right? If you record your sample throughout the whole process. Actually, that brings me to another question that somebody asked me the other day, because they were looking at the recorded sample and they were saying that, okay, I don't see any problems during the sort. And just to remind everyone that when we record, we actually just record a little bit of the sample, but we're not recording during the sort. So during the sorting, while it's sorting, we're not seeing if there's any problem, with fluidics, right? Unless we are recording it throughout the whole sorting process.
And another thing that's just occurred to me as well is that quite often when we're doing the sorting, what we will do is we'll start the sort, and maybe after 5 or 10 minutes, we'll just take a small aliquot and check that the sorting purity is okay. Especially if we've got a 5-hour sort booked, we don't want to get to the end of the 5 hours and find that we've made some mistake in our gating or something like that. So we'll do a quick reality check to make sure that we're sorting what we think we're sorting.
Yeah, absolutely. And it could show you issues like if your laser delay is off, because the window extension isn't very large for sorting, you can actually wind up with issues there as well. So it's always a really good idea. We have another question that popped through from Adam Wright. He asked if we recommend recording throughout the whole sorting process. I think Yeah. So
You
you
can
might want
do.
to take, yeah, you can, depending on how, you know, how long you're sorting for, right? There are some, you know, restrictions you might have as far as how many samples you have to process, how many millions of cells that you have, and the feasibility of it. But you can always take snapshots across as well.
Right. So, Derek, do you want to show the?
Yeah, if you
Yeah.
stop your share, I'll take
Oh, sure.
over and while you do some cleaning.
Mhm.
Okay, so we just wanted to show a couple of slides. Actually, we wanted to show from the first time we did this one, but we kept running out of time. I think today, surprisingly, looks like we're going to be pretty much on time. And that is to do with the checking of the purity that we've just looked at with Rui. So this is a slide I showed 3 webinars ago. And this is what we want, isn't it? This is our unsorted population here on the top 3 panels, and then the equivalent on the bottom of the sorted panel. So we've set a gate around our live cells. We've got a viability diet in there to exclude the dead cells. In this example, we were interested in this double negative population. And then within that double negative population, we wanted to sort this. This is the target population, the eFluor 780 positive. Doesn't matter what the antigens are, but that's the target population. And then at the end of the sort, we can see that we did what we wanted to do. You know, you can see that the purity is 99% and above in each of those boxes. So great that the sorter has done, certainly in terms of purity, has done what we wanted it to do. But it doesn't always work like that, does it? And sometimes there are problems, and sometimes there are many reasons why a sort can go wrong, if you like, in terms of the purity. And sometimes we have to think in terms of machine. Sometimes we have to think in terms of biology as well. So you use just a few examples of that. And this first one was using beads. So we've got 2 different sizes of beads, multiple different fluorochromes in there. But what we were interested in here was just to sort these APC positive beads. So we put our sort gate around the APC positive beads, and the starting percentage is just under 20%. And at the end of the sort, it's 70%. That's not brilliant, is it? As Rui said, we should be able to get 99% purity as long as everything is perfect. But then we have to start thinking about why that isn't 99%. And if we look at this, you know, and as I say virtually every webinar, and I say all the time, flow cytometry is about numbers and visualization, isn't it? And now we know our purity is relatively low. And if we look at why it's low, it's because we've got this population here. Now, this population here isn't in the unsorted population at all. It seems to be a new population that's arisen. So why is that? And with a bit of troubleshooting, what we found is that, you know, we instill upon people that they should clean between their sorted sample and their unsorted sample and then their sorted sample. And that's exactly what the operator had done here. So they'd run some rinse, they'd run some bleach, and then they'd run some water. But what they hadn't done is run that water quite sufficiently, because there was a little bit of residual bleach in the line. And what happened, of course, is our beads have gone through and the first part of that sample has been bleached, so the signal has been reduced. And actually, if we had the time plot here, we would probably see that all these lower APC signals were at the beginning of the data file. So, you know, that's a practical issue that's caused that problem there. What about this situation? Here we've got a sort of CD14, CD16. No, CD14 and 16 are quite often used to delineate monocytes or various states of monocytic development. And what we've done here is 4 sorts, but this one for the CD16 single positive population, this for the CD14, and then this one for this sort of intermediate population. And when we rerun the samples, the single positive and the double positive seem pretty good. And there is a slight movement there. And, you know, as we discussed before, this is mainly due to mathematics. A cell, just because it was inside that box when it was first run, it could quite easily be slightly outside when we rerun it. But this looks pretty good. So does this. But here we're seeing quite a lot of reduction in purity. And actually, this is a biological phenomenon here, because what's happening with these cells is that they're either recycling or internalizing their antigens. And of course, the fluorochrome in that case is either internalized and may lose some fluorescence, or it's shed from the cell. So we end up seeing some cells here that are less positive than we expect them to be. Actually, when they were sorted, they were all in that box. That loss of fluorescence has happened afterwards. And then the final one here is one that we've been talking about today, and the idea of having a viability diet in there. So if we just sort our GFP cells on GFP alone, this is what we're interested in, the GFP positive cells, and you can see they're very well separated from the negatives. Should be the easiest sort in the world. But we rerun it, and actually we find that we've only got 91% purity. Where's that 99% that we're expecting? And as Rui said, not only should we be having a viability dye in our pre-sorted sample, but we should also be adding it to our sorted cells as well. So if we add a viability dye, and in this case we've added propidium iodide, what we find is that these negative cells here are actually cells that have died during the process of the sort. Some cells are fragile. We know that sometimes cells are not happy when they're sorted, and these GFP containing cells have died. They haven't necessarily lost their GFP, but maybe the protein has become slightly less conformationally active. We've lost the fluorescence from the GFP. And once we add that propidium iodide, now when we look at our live cells, we're back to our 99% plus viability. We've still got a few cells here. And this is another sort of anecdotal point, which we might have mentioned before, that you always get slightly better purity if you sort negative cells than if you sort positive cells, because no matter how good our doublet discrimination, there'll always be the odd occasion where we have two cells that are so close together, we can't distinguish them as doublets. And if one of those is GFP positive and one is negative, to the cytometer, that just looks like a GFP positive cell, and it will sort it. Then those two cells end up in the tube, and of course they dissociate. So when we rerun it, we see a negative and a positive. And if I was just sorting this negative population, that would never occur, because I would never sort something that had a GFP signal as well. So for the last few minutes, hopefully we can go back and have a look at the few cells that Rui sorted there with that four-way sort. Remember, we were sorting singlets and doublets, and we're looking at the GFP signal within each of those populations.
We are, yes. We're finding out interesting things that, of course, contradict, well, not everything you said, but no, let me share the screen while I'm recording the one of the post-sort purities. There we go, sharing the screen, everything is here. So as you said, you know, we're sorting, okay, this is good. We're sorting the doublets. We're looking at the post-sort purity check of the doublets for the positive. Try to get at the right windows here. But as you see, we have. I'll just run the last sample, and then we can interpret everything correctly. There we go. And so now I need to just look at the last one.
So Rui, maybe as you're putting on that last sample and recording that, I'll ask a question that came through. So there's a question that came through that says, with the example with the dead cells, would you factor in the dead cells into the percent purity calculation? So because you're measuring RNA, they are still there. So that's a question that came through. I think it could go for anyone, because Derek was presenting the slides.
It's a good question. Yeah, because you know, I think ultimately the sorter has sorted what we asked it to sort, which are things that GFP positive, but obviously ultimately in your tube, you've now got some dead cells. So going forward, if you're taking that to some sort of molecular biology application and you're taking that whole of that population, yes, you do want to know how many of those cells were dead, and actually only 92% of the cells were live in that. So you need to take that into account.
Yeah. Yeah, I would completely agree. You need to factor that in and understand that, you want to make sure that you spin down the cells and make sure that you're really looking at your live cells, your downstream applications, and be careful about how you calculate that as well. And just remember that, you know, how you treat your samples as far as the collection tubes and pre-coating the tubes with serum. And there are certain actions that you can take to keep your cells a little bit happier so that when they are sorted into those tubes, they're not under as much stress as they might be, if they were under not ideal conditions. So for example, if you're working with certain cell lines that don't love to be alone, like many do, or don't like, you can sort into conditioned media. You can make sure that you're utilizing pre-coating of the tubes with serums, that the electrostatic charge doesn't cause for those droplets to really dry up on the side of the tube walls. Be mindful of the volume that you're sorting into, because if you're sorting very small numbers into very high volumes, the cells might not be very happy either. So there are all things that you can do to try and keep your cells a bit happier and reduce the cells that are dying in the process.
Exactly. In that example we showed there, I mean, there was a couple of errors, weren't there, because we didn't have the viability diet in the sort in the first place. So we were sorting some dead cells anyway. And secondly, if you are noticing that 10% of your cells are dying, that probably leads you to try and optimize that in some way, like Kathy was saying, potentially changing the medium that you're sorting into, or treating the cells more gently.
Yeah, absolutely. And there is a question about how you can pre-coat those tubes. So you can either fill them with whole serum, or you could do PBS with 20% FBS, or 5% BSA. You can leave them at room temp for at least 2 hours, or leave them overnight in the fridge. And then what you can do is you can take out and remove the excess serum or collection media beforehand, so that you're just sorting into straight serum. If you used whole serum, some people like sorting into whole serum, or you can have the collection media put in there if you remove that whole buffer that you use to coat the tubes with.
One thing I think we may have mentioned in the previous one is if you are sorting into whole serum and you're going to rerun that as a sort control, be careful, it's full of nonsense.
Yes, absolutely.
Scatter will be all over the shop.
Absolutely.
Okay, so we're ready here to show them what we have here. So again, remember, we sorted these under a singlet gate, the positive and negative, and then we sorted positive and negative under the doublet gate. So what we expect here to see is, under the singlet gate, we should see, a good purity for negative and positive, hopefully. And I think I have a surprise for you. But and with the doublet gate, like Derek was mentioning, we should see the if we're sorting, for instance, for positives, we may have a negative attached, and it will still look positive, right? So we'll sort those. So we expect to have much less purity in that case for the positive than from the negative, because the negative, if there is an aggregate that is a negative and positive, it will show it in the positive gate, and so it won't sort. And it will only sort the aggregates that are both negative or triple negative, etc. Right? So let's start with the singlet. And the singlet, we should have good purity. So here I'm including, because mostly we have singlets once we sorted them, because they essentially break apart as they fall in. So we move the gate, and actually thank you, Fang, for reminding me to move the gate, the doublet gate, to include these. And so, but now we're only looking at the singlet gate, right? And we're looking at negative, so P6 negative. So if we look at P6 negative, we see that we have 82%, a bit lower than what we had with the last sort. But again, we were getting slightly different parts, so maybe we still included some doublets. And I suspect, and this is one of the difficulties with this sample, and that's one of the reasons why we wanted to show you this, is that, in a sample, and actually it would have been nice to show pictures of how this sample looks under the microscope, but you may want to be really restrictive to make sure that you eliminate all of the doublets. So, for some reason, I wouldn't expect any positive here. And we did find some positive cells here, even though we were sorting this gate. Okay, so there's there may be something, I would suspect here, something wrong with the AccuDrop, right? We should look at the timing, the drop charge delay, and just make sure it's correct. Like Kathy was saying, we need to make sure that the drop one is not too far away, and it's not. It's just a couple of 2 pixels away, so it's pretty good. But maybe something to do with the AccuDrop. So that's why also Derek mentioned that it's good to, if you check the post-sort purity right, a few minutes, right before you start sorting, it's important because it will help you to adjust the gates accordingly, right? So in this case, for me, in the first sort, I was tempted to put the singlet gate much more stringent, but in this case, what I'm seeing here is that when I sort for negative and I see positives, that's not a question of doublet discrimination, because we shouldn't see a doublet of a positive versus negative, because that one should fall over here, right? So something to take into account. And in fact, that's why I was saying that Derek, this contradicts a little bit what we would expect, is the positive, so the P5 GFP positive population actually has a pretty good, let me see, P5 97.4, and so I think I was being more stringent now with a singlet gate when we sorted. So this this really helped to get a good purity sort for some reason. Then we sorted the P8 gate. So the P8 gate would be the positive GFP, but the doublets. So again, here we would expect a lot of a not very good purity. And if we include all of the GFP positive cells, we see that it's still 85%. It's not bad, but we do see 12% of negatives. But when we sort the negatives, we shouldn't see any positives at all, right? And again, we're still seeing a lot of positives here for some reason. So again, this similar to this one here, right? That we were seeing still a lot of positives. So something to to take in consideration. I would be tempted here to check the AccuDrop, although it seems that when we sorted the positive, it worked pretty well. Any questions there?
Nope, we don't have any questions coming through at all. It is definitely something interesting that came up, and you can kind of see that there's maybe a slight difference there as well with the viability and how the DAPI looks on the samples with that were supposed to be from the doublet negative, as opposed to the doublet GFP negative compared to the GFP positive doublets. Interesting.
This one compared with this one, right?
Yeah, or maybe just more cells that came through.
Oh yeah. So we had very few of these, as you can see in the actual sample. We had, so if I move this back here, which is what we were sorting, right? We're sorting something like this. You can see that there are very few of these negatives, right? So again, it's interesting that by sorting these, we still capture a lot of these cells here. So when that happens, every time I see positive events when we're sorting negative, that's a good indication that usually it points to the drop charge delay, right? That needs to be adjusted. But on the other hand, we did get good result for sorting positives, right? So if it was a question of timing, well, one thing that it's important to notice is that in the sample here, of course, this would be easy to get good purity, even though the drop charge delay may be off, because we have very few of these, right? So it's very unlikely that it's gonna get contaminated. So that may be the case. And here the frequency is also much lower right? 7
Exactly.
versus 33.
Sometimes you get away with things, don't you, by
Exactly.
luck,
Right.
and quite often you get a good purity, which is great, but your yield is very low, your recovery is very low.
Right.
You could have sorted 10 times as many cells.
Mhm.
And I suppose this is the experience that you get from sorting, and this is
Exactly.
where samples like this that are a little tricky are actually
Mhm.
quite useful for us, I think, because it helps us understand a little bit more what we have to concentrate on and what we have to look for.
Exactly. Yeah, if we had more time, what I would have done now is just go and check the AccuDrop, just make sure that that everything is right, and then just sort again.
And I think this is one of the reasons why we do that sort of quick sort check after 5 minutes, because if
Exactly.
something like this comes up, we can go back and do that, and we haven't lost a lot of time, and we haven't lost a lot of sample.
Very important.
Exactly. Exactly.
Yeah, I like this.
Okay, so we're slightly run over time. So we've got a last couple of slides, I think. If I get my screen share back. Don't see why I can't get it back from Rui, but I seem to not be able to do that. Okay. So hopefully you've all had a chance to ask all the questions that that you want to ask. I think we've covered most of them. There aren't any outstanding ones there. So thank you all for coming. And as usual, at the end of this, we will send you a questionnaire or a link to a questionnaire. Even if you filled that in before, it's quite useful for us to get that information, particularly if you've got any comments or there's anything you want us to see us do in the next few months. And as usual, this is being recorded, so the recording will be available probably by mid next week, and that will be put up onto the OpenFlow Cytometry YouTube site. We're not stopping here. We're going to do something again next month as well. And so we've already got the date for that, which is the 21st of May. So that's another Friday, about 5 weeks away, I think. Same time, 1 o'clock EST and 6 o'clock BST. And rather than using the cytometer, we thought we would look at some data analysis. Now, before we started all of these webinars just over about a year ago, I suppose, no, not quite a year ago, we sent out a sort of questionnaire asking what people would like to see, and we've covered the sort of popular things, but one thing we didn't cover, which is popular, is the idea of data analysis. So we'll be using several data analysis programs well, maybe not all in next time, but we might spread it over a couple of sessions. But we'll look at things like gating and which plots you're likely to use, which metrics you might derive, how you output data that you might want to put into a paper or publication or a lab meeting or whatever. So registration is already open for that. If you don't get to write that down, if you just go to the Crick What's On page, you'll be able to register for that. And I will also be sending this out via the usual social media channels in the next couple of weeks. So this is the very last slide. So if you want to contact us offline, please feel free to do so. You can contact any of us individually here, or you can use the OpenFlow Cytometry Gmail address there. So that will include any specific questions that we haven't managed to answer today, or any suggestions or comments or criticisms, whatever. We can take it all. And of course, follow us on Twitter, or follow MSK and myself on Twitter. Kathy, we're still waiting for you to get your Twitter account up and running, but hopefully by next time we'll
I will
have that
take
as
any
well.
suggestions for clever names, because I just don't have one just yet. It's all
That's
about
the only
name.
thing that's holding you back, right?
It's the only thing.
So if there aren't any more questions, we would like to thank you all for coming. Thank you all for hanging around for 95 minutes today. And we hope to see you on our next webinar in a month's time. Take care, everyone. Stay safe.
Thank you. Take care, everyone.
Bye.
Thanks, everyone. Thanks, Fong.